Hi, I'm here to tell you about a new tool for the detection of contamination in solids and liquids that can also be used to monitor microbial activity. It's called Speedy. It's designed to be used at source, where a sample is taken by industries that need to monitor fluids and macerated solids for microbial contamination. This could be in food and beverages, drinking water, cleaning products, paint and oil, pharmaceuticals, cosmetics, pet food and healthcare products. It's also perfect for academic researchers with an interest in a whole host of areas, from medical to food to pharmaceutical, and as a teaching aid. It's for anyone who needs to understand how microbes behave in a variety of conditions and media. Currently, test equipment varies from large lab-based machines that can test a couple of hundred samples an hour to manual systems like this simple agar plate, a technique invented in 1879, over 130 years ago. No matter what you currently use, Speedy can change the way you work for good. Speedy is a clever little device. It's a microbial respirometer, an instrument that will rapidly grow and monitor bacteria and yeasts in liquid and macerated solids. Let's take a look at how Speedy works. Speedy has two chambers. Inside each one is a disposable sterile culture vessel. A new one is needed for each experiment. The culture vessel is sealed, allowing very sensitive pressure measurements to be made once placed inside Speedy. To run an experiment, you just introduce a growth medium of choice, or use one of our culture vessels that come pre-filled with soluble media capsules. Place the culture vessel into the culture chamber, then press the Start button on Speedy. Simple. Inside the culture vessel there's a paddle mechanism which turns to keep the broth and sample optimally mixed. The paddles are magnetically driven. If microbes are present, they grow and respire, altering the pressure in the headspace of the culture vessel, which is detected by this sensor. This means that microbes are present and growing. Protocols are designed using Speedy software that's loaded onto a Windows PC. While Speedy comes with a set of standard protocols, users can design their own on a PC and download them to Speedy. The variables that make up a protocol are temperature, length of the experiment, and the speed the paddles turn. Protocols can be stored and retrieved on the PC. Once the culture chambers have been populated, Speedy starts to heat or cool and take readings to the predefined protocol. The two chambers can be used to run two experiments to different protocols, or to compare a sample to a control. This animation shows a 500-minute experiment speeded up. Initially, the curve shows the vessel heating. Then, as time goes by, bacteria consume gases such as oxygen, or produce gases such as carbon dioxide, changing the pressure in the vessel headspace, which you can see here as the curve dips, then dramatically climbs. The dip is where bacteria have grown to large numbers and begin to modify the pressure in the headspace. So how long does it take? Well, that depends on the degree of contamination in the sample. For a common organism such as E. coli, Speedy can detect the presence of a single organism in 10 hours. Higher contamination levels will be detected much faster. Where an experiment starts with high numbers of organisms, and the measurement being made is rate of gaseous exchange and microbial activity, results can be generated almost immediately. The brilliant thing about Speedy, apart from its ease of use, is the fact that you can see what's going on inside the culture vessel. Speedy turns microbial activity into data, which means that by connecting Speedy to a PC at the start of the experiment, you can watch the warm-up curve and then see how the experiment's progressing. Speedy lets you visualize your experiment in near real time. Experimental results can be uploaded to a Windows PC by connecting Speedy via this USB port. Alternatively, Data can be transferred via the memory card. So, Speedy is an incredibly useful tool in the factory, on a process line and in the lab. And it's priced for the most cost-conscious industries and financially challenged researchers. www.speedybreedy.com